Okay, my dears. Today we are going to introduce punnet squares, which I'm sure you probably have heard of. And we're going to practice something today called monohybrid crosses. Let me just write that on the screen. Monohybrid, right? Where we have oh, damn, hold on. I'm still getting used to this thing. Obviously, this writing tool right here. It just drives me crazy, doesn't it? Okay, in any case, mono meaning one, right? So we're gonna examine how one trait gets passed down. And right here on the screen, I just want to review with you guys, genotype, right, is the letters, right? A, B, A, A, right? Whereas phenotype is the actual trait. So that would be something like blue eyes, right? Straight hair, etc. Okay, so, right, again, we're seeing, I just wanted to also quickly review our homozygous, heterozygous genotypes, right? So we can see that, let me get this into red. We can see that this capital P is homozygous, meaning the same, right? So homozygous meaning the same, and they're both dominant, they're both uppercase. Whereas this one is also homozygous because they're the same, but they are lowercase, so they're recessive. And this one is heterozygous, hetero meaning different, right? So one of each. Okay, that was just a quick review. So the reason that this exists goes back to this idea of meiosis. So let's break this down a little bit according to Mendel's law of segregation. So what's going on is we have two, this is our cell, right? This is our cell. And in our cell, we have something from dad and something from mom, right? Mom is in blue. And we go through our replication. So we have two of each. We have two of each, right? And then we segregate that out, right? So those divide, right? Note that they're both B's, right? So B is coding for the same trait, just like with the different sizes, right? These are all large, right? So we separate those homologous pairs, and we end up with our 4 gametes, right? Our haploid gamete cells. And then those can be combined, right? So let's say all of these were made in a female. So all of these are eggs, just the female. Then one of these eggs could go over here and meet up with a little sperm. Here's our little sperm. And in the sperm, maybe the sperm has capital B, right? So if this female's egg, capital B, meets up with the sperm egg, then we know, right, the child, after this fertilization, the child will have this genotype, right? Or maybe the little b meets up with the big B, and then the child will have a heterozygous genotype. Anyway, as you can see, this is really complicated, and we don't want to draw this out every time. So what we developed is a way to model this using a punnet square. So instead of doing the meiosis and then figuring out all the possible matches, we model it with a punnet square. Okay, so let's try that. So here we have again, so in parent 1, let's say this is the mom. So we can say in this is the, oh, hold on. In this mom, this is the egg. So the egg could either have a B, capital B, or a lowercase b. Those are the options in the egg for the genes, right? because it only can have one. So these are egg options. Up. Egg options. I know, guys, bear with me on the drawing tool, right? So this could be egg 1 or this could be egg 2. And in the sperm also has options, right? So this is sperm 1 and this is sperm 2. So those are the 2 sperm options. So you can see that we're talking about the same trait, right? We're talking about whether or not the bunny rabbit will be black or brown, right? But we have all these different options. So from the mom is giving a black or a brown option, and so is dad. Dad is giving a black or a brown option. So then here comes the simple part. You're going to combine these. So we're going to do fertilization. So let's say this sperm combines with this egg. These 2 options come together and they form that embryo, that zygote, right? That baby. And then these 2 options come together and they form this possible baby, right? And then these 2 options come together and they form this possible baby. And then these 2 options come together and they form, let me try that again, this possible baby, right? So from mom with these 2 options, right, and from dad with these 2 options, we could get a total of 4 different babies, 4 different options in the baby, right? And from those 4 different options, we need to form our genotype ratio and our phenotype ratio, right? So I'm just trying to explain this within the context of meiosis because Punnett squares is really just a model for what happens in meiosis and fertilization, right? So when that egg, that potential egg, and that potential sperm come together, they fertilize and they make that diploid cell. These are both haploid. Or they make this diploid cell, right? Or they make this diploid cell or this diploid cell. It's a little bit confusing, but we're going to get there. But I think most of you see how we fill out the Punnett square. So the next important thing to do is to understand genotypes and phenotypes, right? So genotypes, again, are the letters, right? So we have to put in our ratio. So we have 1 BB, we have 1 BB, and we have 2 BB, right? So there's one homozygous dominant to 1 homozygous recessive to 2 heterozygous. We can also write this as 25%. How do I get the text to be smaller? That is my question. I wonder if I can do that. I can. Sorry, I figured it out, guys. Right. Yeah, sometimes it takes me a while. In any case, so this can also be 25% BV to 25% homozygous recessive to 50% heterozygous, right? So in other words, when this parent, this mom, and this dad come together and have babies, there's a 25% chance of it being homozygous dominant, there's a 25% chance of it being homozygous recessive, and there's a 50% chance of it being heterozygous. Now, that is the genotype ratio. So the genotype ratio may be the same as the phenotype ratio, or it may not. Remember that the phenotype is the actual trait, right? So whatever this is, like what you're actually seeing, is the phenotype, right? So for our phenotype ratio, we have 1. So let's maybe write in what phenotypes each of these are. So little b, right, is recessive, right? So it's gonna be brown. And big B is both of those are dominant, so we know it's going to be black. That's the only option. Now remember, when we get to this heterozygous, we defer to the dominant, right? This capital B overrides the lowercase b. So these are black. That's the phenotype, right? So in our phenotype ratio over here, we actually have 3 black to 1 brown, or 75% black to 25% brown. Just like that. Does that make sense? Okay, so we're going to try this again on the next page. We're going to do another example. Okay, we're just filling this in, right? Okay. Okay, so this is the Punnett square that's going to appear on your homework so that you can easily type the answers in. So we're going to practice this, right? So in tomatoes, there's a red fruit. R is dominant. So we know that capital R, red, is dominant. Red is dominant and yellow is recessive. So the parent cross is homozygous red. So one of the parents is homozygous red. So we need to write in that genotype, right? So homozygous, we know it needs to be the same, and since it's red, it's dominant, so it's capital R, capital R. And over here, it says the other parent is yellow fruit. And we know that yellow, to be yellow, you have to be homozygous recessive, right? So you have to be little r, little r. So we have our 2 parents. So we're going to put one of our parents on here, capital R, capital R. We're going to put the other parent on here. It doesn't matter which parent you put on which side, but you do need to make sure that, right, one parent, right, this is the options from parent 1, right? That's the only option. And these are the options from parent 2. So they stick together on their side, right? And then we just complete our little Punnett square. So this is going to be capital R, lowercase r. And this is going to be heterozygous, and this is going to be heterozygous, and this is going to be heterozygous, right? We got that from combining these two in fertilization to make this potential egg, right? Remember that these are the egg option, these are the eggs, and these are the sperms, I guess. And these are the babies. Babies, right? We're just exploring the options here. Babies. Okay, so, and then down here it wants you to do your genotype ratio. So we actually have 4 heterozygous, right? 4 capital R. That could also be or 100% heterozygous. Oops. You can also write 100% heterozygous if you don't want to write the letters. And what that looks like with phenotypes, remember what is going to be our phenotype for our heterozygous? It's going to be red because it is that dominant trait that overrides the recessive, right? So we are going to have 100% red or 4 red, 0 yellow. Okay, so that was a pretty easy genotype-phenotype ratio. Notice that it did ask for, right, the F1 generation, which means that these are the parents up here, and F1, remember, is the first generation, right? So we just cross the parents to figure out what that first generation of babies could be. And we know that 100% of those babies are going to be heterozygous. There's no other option. But now we're going to do the next question. So here we are, hold on, with almost the exact same page, but only one thing is different, right? So it's the same exact problem, but we're asked about the F2 generation. So if you remember from the previous page, the F1 is 100% red and 100% heterozygous, which means if we want to get an F2 generation, we need to do an F1 times an F1. We need to cross those two, which is the same thing as saying, and we know that both of those F1s are going to be heterozygous, right? So we're going to do a capital R times capital R, or heterozygous times heterozygous, right? So this is what we're going to cross in our Punnett square, right? Because we're crossing two F1, which we figured out in the previous problem. So mom is capital R, and so is mom is heterozygous, and so is dad, right? Because they have to be. They're 100% heterozygous. And yes, we are crossing siblings here. We're assuming, they're plants. It's not incest. It's fine. Okay, but here we are with the F2, the second generation, right? So we're going to write those in. So here's mom up here, and here's dad down here, right? They're both heterozygous, which we figured out in the previous problem. And then we figure out what that is. So this is, right, so here we have our eggs, we have our sperm, we have our babies. And so one of our babies could be homozygous dominant. One of our babies could be heterozygous. Another baby could be heterozygous. And one baby could be homozygous recessive, right? So down here, we're going to fill out our genotype. So we have 1, capital R, 2 heterozygous, and 1 homozygous recessive, right? Or you can write that as 25% homozygous dominant, 50% heterozygous, and 25% homozygous recessive. And then down here for phenotypes, we can write those in. So if it's homozygous dominant, we know it's going to be red. And if it's homozygous recessive, we know it's going to be yellow. But heterozygous has to be red, right? Because that capital is overriding the lowercase, right? Because it's dominant. It's dominating the yellow, right? There's more red. So our phenotype is just going to be 3 red to 1 yellow, or 75% red to 25% yellow. You can see how it's totally possible to have a different genotype ratio than phenotype ratio, right? So the phenotype might not reveal what's going on underneath with the genes. Okay, hopefully this helps you work on your homework problems, which are assigned. They're all just monohybrid crosses, and this is the thing that you need to fill out. One helpful hint is if there is one of these in the problem, then you should fill it out. You should do a Punnett square to figure out that problem, right? If you see a Punnett square, do a Punnett square. Okay, so that's all for now. Feel free to reach out with questions. It is a little bit difficult, but I think you guys can get the hang of it. Okay.